And Cindia, floor is yours.
Thank you. So hello and welcome to my presentation called On the Road to an Algorithm Model. I'm currently doing my master's internship at Epithalix, and the objective of my thesis is to develop a full primary human cell-based 3D algorithm model. So to do so, we have first to understand how the respiratory tree works in vivo. So the air enters through the nose and the mouth, goes down the trachea, and then the lungs until it reaches the alveoli, where our interest is. Inside the alveoli, there are two main epithelial cell types, which are type 1 pneumocytes and type 2 pneumocytes, which I will call 81 and 82. 81 are in contact with endothelial cells and responsible for gaseous exchanges, and 82 will do secrete surfactants like SPC so that alveolar, the alveoli won't collapse when breathing. Around the alveoli, there are also fibroblasts, which will provide an extracellular matrix. And inside the alveolus will be present alveolar macrophage, which will remove potential pathogens inside the alveoli. So first, we had to isolate endothelial cells, and we did that by isolate them from human lung parenchyma and characterize them for 81 and 82 markers. So we use aquaporin 5 that we can see here and RAGE, which are both markers for 81. And as we can see, they are both positive and colocalized very nicely. For 82, we used HT2 280 and LIDO tracker, which are also positive. So here we can say that we have a population of cells that is positive for 81 and 82 markers. So then we wanted to see the influence of fibroblasts and endothelial cells on pneumocytes differentiation. So to do so, we did three models. The first one here, where we have only pneumocytes on the apical side. Here, a second model where we added fibroblasts on the basolateral side. And finally, a third model where we put endothelial cells instead of fibroblasts on the basolateral side of our inserts. So our first results with the inserts containing only pneumocytes showed by immunofluorescence on day 27 expression of the marker RAGE and HT280. However, we can see that we only have a few 82 cells that are still positive on day 27. And moreover, when we look at the cell's density through DAPI, we can see that it forms aggregations and it is not homogeneous anymore. Concerning the TER, we can see a peak on day 5, and then it decreases and stabilizes through time. So for this model, we can say that on day 27, we only have a few 82 cells remaining, and that the layer of cells on the insert is not homogeneous. However, we were able to quantify SPC through the technique ELISA. For a second model that is made of pneumocytes and fibroblasts, we saw on day 42 a very faint or non-existent expression of RAGE, a nice expression of HT2 280, but we saw that the cells grew smaller over time. And when we look at the cell's density with DAPI, it's very dense. We have many layers of cells, which is not promising to develop an alveolar model. Concerning the TR, it peaks on day 10 and then decreases and stabilizes through time for a very long period of time, as on day 69, our inserts are still alive. However, we saw the presence of mucus and cilia. So on day 42, we measured cilia beating frequency, and we can see that the beating frequency is significant. So from the presence of mucus and the presence of ciliary beating frequency, we can see that there is a deviation of the alveolar phenotype to an upper airway phenotype, which might suggest that our cells are multipotential. Our third model, containing the pneumocytes on the apical side and endothelial cells on the basolateral side, showed on day 35 by immunofluorescence an expression of RAGE where we can see that our cells are round. A nice expression for HT2 280. We have many 82s all over the insert and here the merge. And when we look at the cells density, it's not too dense and the layer of cells is homogeneous. So for the immunofluorescence part, it's very promising. When we look at the TR, it peaks on day 16 and then decreases and stabilizes over time, with the last insert dying on day 45. So on day 20, we did an histological cut section to see better the morphology, and we can see that we have a very thin layer made of 1 to 2 felt thick, which is promising. So for this model, we can say that it lasts through time. We were also able to quantify SPC through ELISA, that we have 81 and 82 present on day 35 and a thin layer of cells. So this model seems very promising for an alveolar model. So we wanted to go further and did transmission electron microscopy. We sent our inserts to l'Université de Tours, to Julien Gaillard. So we could see how it looked like on day 20. So here we have the inserts. There we have a first layer of cells made of type 1 pneumocytes. And here we have a second layer of cells made of type 2 pneumocytes that contains the characteristic lamellar bodies. So we wanted to go further with this model. So we took it, added macrophages on day 19, and did an overnight incubation. So we obtained this model that was made of pneumocytes and macrophages on the apical side and endothelial cells on the basolateral side. And then we wanted to see if those macrophages were functional. So to do so, we added diamazone to test the phagocytosis ability of macrophages. Indeed, diamazone is a complex that is taken from yeast and that is coupled with a fluorochrome that is sensitive to pH and will turn red when in acidic compartments. So it means that if macrophages phagocytose them. Our results on day 20, we can see here our macrophages on the phase contrast, and here the diamazone that was phagocytosed by the macrophages, so positive, and our negative control. So we can say from that the macrophages that we added on our 3D culture are functional. So in conclusion, I did a summary table with our 3 models and the main characteristics for each. And the key points of my presentation are that first, pneumocytes were isolated from human lung parenchyma and that they seem to be multipotent progenitor cells. Second, that our data suggests that fibroblasts may be detrimental and in the endothelial cells beneficial for long-term maintenance of the alveolar barrier and its phenotype. Also, as of today, it is possible to make an alveolar model that is functional up to 35 days, that shows 81 and 82 markers, has an homogeneous morphology that produces SPC, and is able to maintain a barrier function through time. Finally, the last point is that we can prepare a fully primary alveolar endothelial and macrophage model. Thank you for your attention.
Thanks a lot, Cindia. Again, your first presentation, congratulations. We have a panel of questions again, so I will need to select just few of those, and you would have to answer in the Q&A session afterwards. So I will pick up some of them. So the first one is, thank you for your presentation, very interesting. How can you explain the increase of TIR until day 16 and then the decrease of it? Is this decrease normal?
Well, first it will increase as the cells will go on and proliferate, and then it will stabilize. It will decrease when it stabilizes, and the tight junctions are formed. So it's normal that then it decreases after a peak.
Okay. So the second one is what type of macrophages are you using in the alveolar co-culture?
The macrophages we use are alveolar macrophages that we isolated from human lungs, and then we are able to freeze and store.
Okay. Another question: have you used primary fibroblast endothelial cell for your model or cell line?
So all cells are full primary human cells. So no cell lines were used to do these models.
Okay, I think it's the last question that I have in this Q&A. Is that, do you ship the kits?
Not yet, but we still have to do some experiments to standardize, but it's something we plan to do.
Okay, so if now the chairs have other additional question.
No, no more question now. I will give the floor to Professor Sarah Cherry.